Hey class, how are you doing? Um, I realize I say that every time, I'm sorry. I'm probably not going to stop though. So here is our nervous system lecture. So structure and function of the nervous system, our goals for what we're trying to get over, at least within an exercise context. First off, look at the structure of how the nervous system works. There's effectively two different divisions, and then it subdivides even further. Um, and then looking at parts of a neuron, so dendrites, axons, soma, that type of thing. Um, and then go into action potentials. How do we even propagate information? Then we're going to talk about the sodium-potassium pump, how that works, some proprioceptors, and there's really just two main ones that I really want us to understand: the muscle spindle in regard to the stretch reflex, and the Golgi tendon organ in regard to autogenic inhibition. Then after that, we'll talk a little bit about motor unit recruitment, the size principle, these things, and a little bit on reflexes, a little bit on how the brain works, and potentially some other stuff going on. So right here, starting off, general nervous system functions. It effectively controls the internal environment, probably one of the most important systems that we have, because it sends signals and messages to virtually every other system. So the endocrine system wouldn't know what to do unless the nervous system effectively told it to. Fairly similar thing with skeletal muscle. But here, control of the internal environment, voluntary control of movements, so working your hands, your legs, your back, moving your neck back and forth, whatever, all of that. Programming spinal cord reflexes. So if you've ever if you've ever touched a really hot stove and then retracted your hand very rapidly, that's a spinal cord reflex, and the nervous system is functionally responsible for that. If you had a problem in the nervous system, it couldn't do that. And potentially, maybe the most important assimilation of experiences necessary for memory and learning. So hopefully right now, everything that I'm saying is kind of going into your brain, and then you're trying to make some connections between different aspects of information so that you can perform well on it on a test, but also later on in your life, just kind of generally knowing what's going on. Now, the organization of the nervous system, two primary parts: CNS and PNS, so central nervous system, and this is all central in regard to your body, so the brain and spinal column, and then also the peripheral nervous system. So that effectively just means any type of neuron that is outside of your central nervous system. So those go into two different divisions: the sensory and motor division. So frequently the terms that we use here are afferent and efferent, and I'll say them that way so that it makes it a little bit more obvious. So afferent is going sending signals towards your central nervous system. So it's picking up information from outside, sending it back in, so that your central nervous system kind of knows what's going on. Now, the motor or efferent division, these are neurons or neuronal fibers that are sending information from your central nervous system out. So effectively, it is in effect just like that. Now, something kind of interesting here. Typically, how we understand it, there is a little bit more information coming out on this, but more or less the central nervous system doesn't really repair. So once we have a damage or some type of insult within the central nervous system, it either doesn't repair or it takes an absurdly long time for it to repair. Now, the peripheral nervous system actually can repair itself, and whenever we look at the length of an axon or a dendrite, or different projections from a nerve cell called a neuron, it seems like they can repair themselves by approximately 1 millimeter a day on average. So that's if you cut some place in, like, say, your finger, and you hacked off like a part of an axon, it is possible that like it's going to come back. Well, like the neuron, the axon. Not if you chop off your finger, it won't come back. We're not lizards. But there we go. Now here, these anatomical divisions, and this is kind of how I want you to see it. It subdivides even further. But here, there's the brain, the spinal cord or spinal column, however you want to call that. That's the central nervous system, and then everything outside of that goes into the afferent and efferent divisions. So the afferent, that's like somatic, so kind of like your body, like your I don't know, sense of touch and things, visceral, special sensory, but really the ones that I'm a little bit more concerned with, at least in regard to exercise, are these efferent ones. So somatic and autonomic. So somatic meaning that, this is where we have conscious control, this efferent division, and then further into the somatic motor aspect of the peripheral nervous system, that's where muscle contractions actually happen. That's our control of that. Now, autonomic, effectively meaning that we don't have control of it right there. That would be sending different neurotransmitters out to either like turn you up or turn you down. So if we think about, say, your heart rate, typically, if there's no impact of the sympathetic or parasympathetic nervous system, your heart rate will beat at about 100 beats per minute. And if there's parasympathetic stimulation, it will go lower. If there's sympathetic stimulation, it will go higher. Broadly, we think about those as sympathetic being the fight or flight, parasympathetic rest and digest. There's also the enteric one, which quite confusing. So I'm not going to talk about the enteric one much, if at all. Now here's just more or less a relationship and mapping a few more things onto here that I think it's important for us to know. So we've got nerves going everywhere in our body. Like, luckily, this isn't an anatomy and physiology class making you memorize all of the cranial nerves. I'm sure many of you have had to do that before. It's quite difficult. But I want to guide your attention to over here, where all of these things are coming from. So sensory receptors. So this can be like a chemoreceptor, meaning that like we feel some type of chemical buildup in a muscle, and that's going to send some information back to your peripheral nervous system and then subsequently to your central nervous system. And say if there's a buildup of carbon dioxide or hydrogen, that means that there's a greater amount of metabolic activity in a certain area. And then that will potentially send some type of signal out, potentially through the autonomic nervous system, to open up smooth muscles, like have vasodilation, so we can get more blood flow to a skeletal muscle, or what have you, right? So this is more or less how this works. So sensory signals coming in through, and then sending signals back through this motor division. And somatic, we broadly think about that as skeletal muscle, and autonomic as smooth muscle. Think about that as like blood vessels. So opening up like arteries, arterioles, capillaries, things like that, so that we can get more fuel and nutrients, traveling in our blood to certain areas. Also, cardiac muscle. Cardiac muscle is not somatic. You can't control your left ventricle to make it contract harder. But you can start exercise and like it will contract harder, but that's through an autonomic means. And then glands. So the whole endocrine stuff that we were talking about in the cell signaling and hormone chapter, right? So go ahead and read through those summaries, and then we'll move on from there. Now let's look right here, the structure of a neuron. There's a couple of different parts that I want you to know. So first off, being the cell body, this is frequently called a soma, which is spelled S-O-M-A, soma, and that effectively contains the nucleus. So this is more or less the um, uh, most central aspect of a neuron, and a neuron is just a nerve cell, right? And basically, the soma or the cell body does all of the calculations about what signal to send out. But where it gets the signals from, like its idea of what to do, it gets this from dendrites. So dendrites conduct impulses toward the cell body. So effectively, soma has dendrites connecting to more upstream neurons, and then it's going to be receiving information there. Now the cell body will propagate or send down a signal through something called an axon. So this carries electrical impulses away from cell bodies. And they're covered by something called Schwann cells, and they have myelin sheaths on them. And I'll talk about that like ever so slightly, but really not too much. And then there's a synapse or synapse. I believe synapse is the right pronunciation, but I'm sure most of you know that I don't pronounce words very well most of the time. But synapse is a contact point between an axon of one neuron and a dendrite of another neuron. So overall, this is more or less how it looks. So here is a nerve cell. It has all of the fairly interesting stuff in it. It's got mitochondria, Golgi apparati, a nucleus, nissal substances, which I'm not really going to talk about. There's an axon hillock, and all sorts of other things. So here's the dendrites up here. It would be receiving some information. It would be doing some flavor of calculation and then sending a signal on down. So in the most simple sense, here's how we could think about it. Like you decide to contract your bicep for whatever reason, and then there's going to be a signal to a nerve that, hey, let's have a contraction. Then a nerve is going to be like, oh, okay, we should have contraction. And then there's going to be a signal down an axon where these aspects of the axon are going to effectively connect to a skeletal muscle, and then you'll have a contraction. Now, something fairly interesting here, these myelin sheaths. Whenever we're sending a signal, if we have enough like myelin sheaths, we can actually propagate a signal quite a bit faster. So how I was taught it, if you practice a given motor pattern, say like a bicep curl or throwing a curveball or whatever, every time you do that, then you'll add another layer of a myelin sheath, which a myelin sheath is really just this insulating, fatty, cholesterol-laden substance that makes it to where an electrical signal will go faster down an axon. So the more myelin sheaths you have along an axon, the faster you can send a signal, right? So another particular thing that I think is fairly interesting is this axon diameter. There are differences between like slow and fast twitch muscle fibers with how big this axon diameter is. So the axon diameter is actually quite a bit larger for fast twitch muscle fibers, so typically innervating like larger muscle fibers and things like that. So just a couple of tidbits of information for us there. Now here with synaptic transmission, here we already talked about dendrites, the soma or cell body. I think everyone can tell I prefer to call it a soma. It's faster. Dendrites pick up information, send it to the soma, then down the axon, and then so on and so forth. Now, with this synapse right here, the presynaptic one is, well, really the one propagating the information, and the one receiving the information is the postsynaptic neuron. Now, this postsynaptic neuron will become a presynaptic neuron whenever it propagates information to another neuron. There's numerous neurons longitudinally throughout the body, like sending one signal to another, sending one signal to another. It's kind of like playing telephone. And yeah, no, I think that's at least a funny way to explain it. Whenever you think about how telephone information always gets messed up, and the information frequently does get a little bit less clear the further it goes down the peripheral nervous system. All right, so here, let's think about neurons in a couple of different ways. So starting off, they are called excitable tissues, meaning that they're irritable and they conduct information. So anytime, and this is about to be one of my bad jokes, so get ready for this. Gosh, I do this every year. All right. So if anyone ever says that you're irritable, all that actually means in a neurophysiological sense is that you have an ability to respond to a stimulus and convert it into a neural impulse. So just about the only things that aren't irritable are rocks. So if anyone ever calls you irritable, it's because, well, you're not a rock. You're a human. So there we go. I'm sure no one is laughing. So that's good. But irritability, meaning that neurons, they can respond to stimuli and then do something with it. And also they conduct information. So they transmit this impulse along an axon or further down a particular cell. Now here, resting membrane potential, there's a few things that I want us to know about this. Is within a neuron, there's typically a negative charge within a cell. It's normally shown as around negative 70 millivolts or so, negative 65, negative 55, depending on what textbook that you look at. I don't really care if you know exactly the value for it, but we have a negative charge within a cell, and what it's determined by is the permeability of plasma membrane to different ions. So the main ions that we think about are potassium and sodium, and we're going to have to know which ones are primarily extracellular and intracellular going forward from here. But the resting membrane potential is maintained by something called the Sodium-Potassium Pump. So within a neuron, and actually within a skeletal muscle, this is mostly what is maintaining the charge, like at a particular at the particular negative charge. So here, potassium tends to diffuse out of a cell, and sodium-potassium moves 2 potassium in and 3 sodium out. So that's a net of 1 positive going outside the cell, which is maintaining the negativity of it. So here, this is more or less what's going on. Now, something that I don't think enough people talk about is why is the inside of a cell, in particular like a neuron or a skeletal muscle cell, a myocyte, why are they negative? And hopefully through talking about like AKT, mTOR, AMPK, all of these different enzymes, phosphorylation cascades, remember like PO4 negative, right? Why that it's negative, these fixed anions, this is because mostly the proteins within a cell, and proteins being, you know, enzymes, different cell signals, what have you, mostly these proteins are negatively charged. So that is really what's driving the negative charge, or the charge of a particular cell the most. So here, typically, this is the directionality of these if there wasn't a particular like gated channel. So here let's look at this. The 2 main things that I want us to know. So there's various intracellular fluids and ions, extracellular fluids and ions. So what I want you to get from here is sodium is the primary extracellular ion and potassium is the primary intracellular ion. Now, chloride, being negatively charged, is mostly follows where sodium is. Now there is some within the cell, and not potassium, sorry, calcium. Calcium being a doubly charged ion. That one is a little bit more in the extracellular fluid. Those two I don't find as abundantly important for us to be aware of, but really these two, sodium and potassium, being really important for us to know. So sodium extracellular, potassium intracellular. Na out, K in, right? Now, there is this concept of an ion-gated channel, meaning that there are these particular proteins that more or less function as pores that are controlled by different mechanisms to either be open or shut. So once they're open, in the case of, say, an action potential, say, they will open up, and then different ions will flow down a concentration gradient. So, like, take sodium, for example, being a primary extracellular fluid. Extracellular ion. Sorry, it's early today. Being a primarily extracellular ion. Once these channels are open, then it will flood into the cell, the neuron, and that will change the charge of the neuron, the intracellular space, from like a -65 to something fairly positive, at least in terms of integers, like quite positive. And then that can subsequently send a signal down the axon further. Now here, this is something that I want you to know really well. Let's start at 1. So the sodium-potassium pump, the Na/K pump, as I like to think about it. So what's going on here, this is happening virtually all the time, but primarily at rest, around homeostasis, trying to maintain the negative charge within a cell. Now, it is this is called active transport because it actually consumes an ATP. So this is happening within a neuron and within skeletal muscle as well. There's in the next chapter I'll talk about the 3 main areas where skeletal muscle is actually using ATP, and the sodium-potassium pump is one of the main ones. But here let's talk about this. So first off, the sodium-potassium pump, which is just a protein lodged in the cell membrane of the neuron or skeletal muscle, whatever cell type you want to think. So carrier has a shape that allows it to take up 3 sodiums. So 3 sodiums go inside, and they made them square, so they go into these square little pegs. Kind of cool stuff. We put an ATP in the side of it, so the ATP is split, and a phosphate group attaches to the carrier. So that phosphate group that alters the charge of the sodium-potassium pump, and then that charge, like a magnet, is going to change the shape of it, and then it's going to open up to the outside, and then the sodium is going to go into the extracellular space. So that change of shape results and causes the carrier to release these sodiums, and while it's open, the potassium is going to flow right into there and effectively from that the potassium coming into contact with that's going to then again alter the charge of this, and then that that phosphate, get all this straight, that phosphate is going to be released, and then the potassium, the K, is going to be put into the intracellular space. So that's how a sodium-potassium pump works. So make sure to read your book with all of this. It's fairly interesting stuff. All right, now let's talk about action potentials. So this is effectively what is happening whenever there is actually a signal to be transmitted. And just to remind you, I want you to think about this. as sending a signal for a muscle to contract. So that's really the main thing that I want you to know. So it occurs when a stimulus of sufficient strength, and there's a couple of different ways how we can do this. So we have spatial and temporal summation. I'm going to talk about that here in a little bit. So these ion-gated channels open for sodium, which is the extracellular ion, and then that diffuses into the cell. So the inside becomes very positive, which alters that negative 65 resting membrane potential to go up quite a bit to like fairly positive values. Like like we'll talk a little bit more about that. And then after that signal is transmitted, it will return back to the resting membrane potential through a mechanism of potassium leaving the cell and the sodium channels actually close so they can't come in anymore. Now here's a particular thing that goes along with the nervous system and also skeletal muscle. So we have something called the all or none principle, meaning that like an action potential can't just kind of go. It's either on or off. It's like a switch. Like it's like a light that you turn on the switch and it's on, you turn it off and it's off. We don't have a dimmer switch, meaning like there's various shades of light. Shades of light? I don't know. However that works. Various levels of light in how a muscle contracts or how a nerve sends a signal. So like I'll get into how muscles actually regulate force. It's typically through getting more muscle fibers to contract, but like one muscle fiber can either contract or not contract, just like a nerve can only do a signal or not do a signal. So it effectively looks like this. So we have a resting membrane potential and say here's the onset of a stimulus. So a dendrite is accepting some information, telling it to go, and then the soma has to think about it a little bit, and then it's going to go and send the signal, and that's what we have in action potential. And we have to reach a particular threshold, so we have to have a certain amount of sodium get into the cell to get it just positive enough. And then once it's positive enough, then the channel will open quite a bit, and then there's just a flood of sodium into the cell. And then we're going to propagate a signal, meaning like a signal to contract, say. And then we'll have repolarization, meaning getting more negative, and then we'll be back to normal, more or less. So this is functionally how it works right here. So potassium being inside of a particular cell. And really how I like to think about this is an axon sending information to a skeletal muscle that's over here, and over here being the soma, or a dendrite or whatever, that we functionally have to send the signal on down. So whenever we get the threshold stimulus, getting enough of a signal to go, then we're going to depolarize, meaning get more positive on down the axon just a little bit at a time. And I mean, this happens fairly fast, but hopefully everyone is seeing what I'm trying to explain right here, that like here's the axon that's closest, or part of the axon that's closest to the cell body. Here's the part of the axon that's closest to the synapse with the following dendrite or skeletal muscle. So it has to be sent down like that. So here we have like sodium channels that are open, potassium channels that are closed, so that all of them have to be in there so that we get a much more positive charge. And then down here, the sodium channels are going to close, then we're going to have certain potassium channels open so that the potassium goes out, so that we're effectively getting less positive. So that would be repolarization. So depolarization, more or less getting more positive, meaning that the difference between the extracellular charge and the intracellular charge aren't that different, and then repolarization is making them different again. Hopefully that makes sense. Now, within an action potential, there's two parts that I want to talk about. There's an absolute refractory period and a relative refractory period. And this actually has quite a bit to do with muscle contraction and reaction time, really. So within an absolute refractory period, no matter how much of a signal that we get from another neuron, we cannot send another action potential sequence or signal. So whenever we get the signal to go, once we get the green light on that, then it's going to go, but like once we hit a certain point after the absolute refractory period, this relative refractory period, it is more difficult to send a signal, but if you have a signal of sufficient strength, meaning coming from numerous neurons, then then we can actually get another signal to go. And there's an interesting thing with, there was an old study with like bathing neurons in higher or lower levels of testosterone, and I believe this is right. It's one of these two. I'd have to go back and look it up, but it's either compressing the absolute refractory period or the relative refractory period. I think it's the absolute refractory period that actually gets compressed so that we have less of that, so that we could send another signal a little bit faster. So higher levels of testosterone don't always lead to higher muscle mass, but it can enhance reaction time. That would be another aspect of it, and that's really just for your information. I'm not sure how important that is for us going forward. So now, neurotransmitters and synaptic transmission. So synapse, or synapse, sorry. Just a couple of things about this. So a small gap between presynaptic neuron and postsynaptic neuron, like where the axon and dendrite are meeting. There's actually no physical touch there. There's just a little bit of a space where typically either acetylcholine or norepinephrine are transmitted across. Typically, acetylcholine is the one that we're most worried about, especially with the somatic division of the efferent. But yeah, the neurotransmitter, it's just a chemical message released from the presynaptic membrane, and it binds to the postsynaptic membrane, and then that's more or less what causes the depolarization. So it works in various ways like this. There's a whole lot of memorizable parts here, which I'm not really gonna have you memorize, like this infoot or terminal bouton. I believe it's pronounced in somewhat of a more French way like that. Now here, there's this synaptic cleft where we have the neurotransmitters actually transmitted. Not abundantly important there. In skeletal muscle, we're going to talk about like the importance of acetylcholine in this place, but so far, not so much. Now, how these work. Neurotransmitters and this whole synaptic transmission, we have two different ways of, we have excitatory postsynaptic potentials, meaning that it's trying to cause depolarization, so trying to get them more positive. And then there's inhibitory ones, which are functionally trying to get them more hyperpolarized. And there's effectively this EPSP is the signal of go, and this IPSP is the signal of stop. And there's two flavors of the signal go. There's temporal and spatial. So I want you to understand this in a way that a temporal summation is a signal from a singular neuron numerous times, sending numerous go signals. So numerous EPSPs from one neuron. Now, a spatial summation, this is a go signal all at once from numerous different neurons. So kind of think about it like this. This is one guy telling you to go, and then telling you to go again, and then telling you to go again, and then again, and then again. Now this spatial summation, this is like a whole group of people telling you to go all at once. And both of these are ways of propagating a action potential and getting us really depolarized, but that's at least how I think about it. Well, they can cause depolarization. They won't always, because they're competing with IPSPs, which are effectively the no signal. So if we think about it like this is kind of how it looks, getting us to a particular threshold. Say if we have one EPSP here, and we don't get to the threshold, then nothing happens, another EPSP, and then we don't get to the threshold, nothing happens. Now this would be a temporal summation. So one neuron telling you to go, and then once that second one hits you on the downturn, then you actually get to it, get to the threshold, and then send the signal to say contract. Now spatial summation, it's two going at once, which would be enough to get us to a critical threshold. Now this one right here, an IPSP, that's getting us lower, so getting us more negative, for example. But this can happen quite a bit, like we have an EPSP and an IPSP, both hitting us at once, and then virtually nothing happens. So if anybody ever feels indecisive about something, well, sometimes your nervous system can be indecisive. Like you can have like one go, one no signal, for example, right? And here's just another way to look at it, how I like to look at it, like an EPSP, IPSP, two EPSPs getting us to an action potential, and really just think about this as the more or less, I don't know, chemical charge way of thinking about how we actually contract muscles. So just more or less kind of like some basic stuff going on right there. All right, now let's talk about some sensory information, the afferent aspect of the peripheral nervous system. Sorry, I got some in my throat, geez, come on. All right, and some reflexes. So we have something called proprioception or proprioceptors. So all that these are receptors that provide the central nervous system with information about body positions. So they're frequently located in the joints and muscles. We're really going to be more concerned with the muscle ones over the joint ones. I probably won't test on the joint ones. We also have this other term called kinesthesia, which kin just means movement. So it's really just the conscious recognition of position of body parts and limb movement rates. So I don't know, whenever I think about kinesthesia, I always think about people in like the X Games or gymnasts who are like hopping up in the air doing like 900 on a skateboard or multiple flips, and they know exactly where their, I mean, more or less subconsciously know exactly where their limbs are at any given moment in time to like extend their limbs or retract their limbs so that they can alter like angular velocity rates. I don't know. I'm sure in biomechanics classes that you talk about those types of things. But here, joint proprioceptors, we have three that are kind of going on. So we have free nerve endings. So these are sensitive to touch and pressure. They're initially strongly stimulated, and then they adapt. Kind of these free nerve endings, like whenever you first put on, I don't know, a shirt or something, if anybody has, what is it? Like a tag in the back of your shirt. So you'll really notice it, and then after you have it on for long enough, then you'll effectively adapt, and you won't even realize that you're wearing a shirt or the tag's touching you or what have you. Then there's these Golgi type receptors, which are similar to free nerve endings, but they're in ligaments and around joints. Now this Pacinian corpuscles, I'm Sure, I said that incorrectly, but that's okay because I'm not going to test on it. These are in tissues and around joints, and they really just detect like angular velocity and joint rotation and things. So that's effectively all I really want you to know about joint proprioceptors. Now for muscle proprioceptors, quite a bit I want you to know. And there's a lot about these muscle spindles and Golgi tendon organs that I want us to be aware of. So overall, what these are allowing us to know is the muscle length and amount of tension developed on a muscle. So overall, like here's what I want you to know. Muscle spindles, they're sensitive to length, meaning like if a muscle gets too far extended, then it's going to send a signal to contract harder. So like it gets too long and it needs to contract to protect itself. Now a Golgi tendon organ is a little bit different. It is detecting tension or force, and that tension that it's detecting, it will effectively send a signal to relax so that we don't develop too much tension and effectively damage the body. So overall, like muscle spindles will detect length and send out an efferent stimulus to contract hard. And then a Golgi tendon organ will detect tension and send out a signal mostly in IPSP to stop contracting. So muscle spindle, length, contract hard, Golgi tendon organ, tension, stop contracting. So the muscle spindle, let's talk about this just a little bit. It's associated with a stretch reflex, like the knee-jerk reflex if you've ever been hit in the patellar tendon. What that's doing is that is acutely lengthening your various quadricep muscles. And those sarcomeres are going to be expanded a little bit, and then that's going to send a signal just to the spinal column to make it to where it contracts really hard, trying to protect the knee joint. So there's a few things in here that I want us to know about. So there's intrafusal fibers, which these run parallel to normal muscle fibers, which are called extrafusal. I don't think that that's super important, but this is a little bit important if you really want to get into the weeds of the nervous system. What's actually stimulating our skeletal muscles to contract, like your biceps, quads, whatever, is an alpha motor neuron, but the muscle spindle is actually stimulated by what's called a gamma motor neuron. So pretty cool there. Now the stretch reflex is frequently what's associated with the muscle spindle. So that is stretch on a muscle causing a reflex reaction. So really overall the meaning of a reflex reaction is just that the signal is basically going to just the lowest aspect of your central nervous system, your spinal column first, and there is a signal on up to your brain, but it's acting in a very fast sense, so it's almost unconscious in the way that it works. So this is kind of what's going on here. Like, read through this. I think that this is very cool for us to know. So say there's a sensation in these intrafusal fibers that we're getting a little too long, and if we get too long, that's going to damage the muscle or the joint or what have you. Then there's going to be a signal back to the spinal column, and that will effectively stimulate a motor neuron to contract in that muscle, functionally protecting this joint and that muscle. Now that signal is going right through the spinal column. If you can't tell, like the dorsal and ventral root of the spinal column, that's kind of how that's going. It will send a signal back up to the brain, but you'll actually contract before you even consciously recognize that it's happening. So read through this, make sure that you understand how this muscle spindle is working. Now let's look at the Golgi tendon organ, frequently called the GTO. So this monitors tension development in the muscles. So how I think about this is, if anyone's ever, I don't know, maxed out on a deadlift or something, and then all of a sudden your like hands just let go because like you're grinding on it and it's really heavy, That's more or less the Golgi tendon organ telling you, nope, don't. Because if you keep on going, you're going to hurt yourself. This stimulates an IPSP, which leads to relaxation of the muscle. So this one being a lot of IPSPs going on. So it's now something very interesting here. We'll talk about strength development later on, and partially what strength development is desensitizing the Golgi tendon organ. So whenever you start, like say lifting weights, doing push-ups, breakdancing, like whatever people do, they will effectively, like their body will be fairly conservative in what it thinks that it can do and not die. And then after lifting something or doing things numerous times, then the Golgi tendon organ will become less sensitive. And this is really what, if anybody's ever done like plyometrics or depth jumps in particular, like jumping off of a high box, hitting the ground, and then jumping up, that's really what you're trying to do with that, is you're trying to deaden or silence the GTO so that you can actually fully contract a muscle without any what's called autogenic inhibition. So autogenic inhibition is what's associated with the Golgi tendon organ. Like here, let's look through this. So sensory neuron, a lot of tension development, then it's going to send the signal up here that, oh man, too much tension. And then there's going to be something called an inhibitory interneuron, so between two different neuron types. And this is essentially going to send a signal stopping the alpha motor neuron from contracting. Now, there will be a signal up to the brain, but if anyone's ever maxed out on deadlift or whatever, you don't realize that you're going to completely not get it until you don't, I guess. So there we go with that. All right, now let's talk just a little bit about muscle chemoreceptors, so meaning chemical. The main ones that I want us to know are hydrogen ions and carbon dioxide, which are both indicative of lots of metabolic activity happening in the muscle. So these chemoreceptors are located, in and around muscles, and will effectively detect how much metabolic activity is happening within that particular muscle, and that's going to send a signal afferently back to the central nervous system, and then the nervous system is going to send a signal through the autonomic division of the, here let's think about that. Well, functionally send a signal through the efferent, autonomic division to open up blood vessels, and frequently will send out more neurotransmitters or hormones to direct blood flow or fuel to a particular area that has a really high metabolic demand. This is why, or at least part of the reason why, whenever, say, you're doing bicep curls and then you feel something called the pump, which in scientific terms is just called transient hypertrophy, that the blood vessels are opening because the nervous system got a signal that, hey, there's a lot of hydrogen ions and a lot of carbon dioxide being produced right here, so we better open up the pathways to get to that area so that we can get more fuel there, because that's where the need is. So really just basically how muscles work is a needs-based system. So if you're working your calves, say, we're not going to open up the blood vessels going towards your forearms. So that's kind of how that works. And these chemoreceptors, really just about metabolic rate, is mostly what those are doing. So now let's talk just a little bit about somatic motor function, and probably the biggest thing here is going to be a motor unit. This is a term that you will always need to know, I believe. Here I'll backtrack to the first point, I guess. Somatic motor neurons of the peripheral nervous system, these are responsible for carrying neural signals from the spinal cord to skeletal muscle, right? So spinal cord to skeletal muscle. Now, we have these things called motor neurons, which is, a motor unit, which is just a motor neuron and all of the muscle fibers that it innervates. And there's a particular idea here that I want you to understand, is that a particular motor neuron, an alpha motor neuron, for skeletal muscle, it is going to innervate a certain amount of muscle fibers. And we have things that have low ratios and high ratios. So a lower ratio would be, say, some muscle fibers in, like, say, your hands and fingers. So we have a lot more control for like fine motor tasks, like you can pick up pencils, you can I don't know, like text or whatever. We can type various interesting things, and that's due to a low ratio of like muscle fibers to the motor neuron. Now we have other muscles that have higher ratios, so like, say, your quadriceps, glutes, biceps, things like that. We don't have a lot of fine control in those because effectively we just need it to either go or not go. And this is one of the reasons, at least how humans evolved, that I think is fairly interesting. Why, say, chimpanzees are a lot stronger than us, because their ratios on average are much higher. Like they can't, like, paint, for example. Now they can squeeze their hands really, really hard, much harder than any human could, by I don't know, like an order of magnitude probably. But I don't know. I just think that that's interesting, that these low ratios are effectively why all of us are able to do most of the things that we can do. But like these higher ratios, well, yeah, there is something that, like, potentially faster twitch muscle fibers probably have slightly higher ratios. So something like that. Now here, this being a motor neuron innervating numerous muscle fibers, this is kind of just showing how this works. And all of these guys within here would be collectively called a motor unit, because it's a motor neuron and all the muscle fibers that it innervates. That's more or less how that works. Now, we have something, how we actually recruit different muscle fibers, or actually upstream different motor units. So here's something to know. The muscle fiber is actually dictated by the type of motor unit that innervates it. So we think about type 1 or slow twitch muscle fibers. They are actually innervated by type S motor units, and then type 2A muscle fibers are innervated by type FR, fast fatigue resistant, that's what that means, motor units. And then our fastest twitch muscle fibers, type 2X, it's not type 2B, everybody, type 2X, are actually innervated by type FF. So I would know this. And like the axons of these go from smallest to largest, and that's functionally how that works. So we have something called, like, the size principle, right? So recruitment of more muscle fibers through motor unit activation. So that's motor unit recruitment, sorry. But the size principle is we actually innervate these from smallest to largest, and this makes sense. We only need to, we never want to do more work than we have to. So if small ones will activate a muscle, we'll use those. And we will only use larger ones if we must. Basically, whenever we're contracting as hard as we possibly can, we're actually contracting all of our slow twitch muscle fibers too, or our type S motor units are being active. Now, it actually has to go this way, because in order for these to reach a threshold for an action potential to be propagated, the threshold for type S is actually lower than type FR, and that's lower than type FF. So this is how this recruitment pattern always works. Always. So type S, type FR, type FF. I used to think that potentially maybe we could hop to the type FF if we activate something called a Renshaw cell, but I don't think that that's very true. So here, this is functionally how this works. So percentage of max force. So think about this, 100% of what you can bench press, curl, breakdance, whatever, versus lower percentages. We will recruit more and more the more that we need, basically. So that's our type S, type FR, type FF. And hopefully this graph makes sense to everybody in here. Here's another way to think about it. Like I like this because this is more or less showing the diameter of the axon. So smaller motor neurons with smaller motor units, and then larger ones with more force being required there. And this is just a picture I like to show a lot of people, that really if we're trying to get all of our motor units, and all of our muscle fibers to be active, it's actually probably the smartest to lift fairly heavy or to have high force production. So like if you're doing a 1 repetition max of whatever, something, or if you're jumping, plyometrics tend to like activate just about everything, you're actually getting all of these lower threshold motor units and all of these smaller muscle fibers. So that's something to like just pay attention to. Now, does that mean that like all endurance athletes who are mostly active down here should just max out on squats and deadlifts all the time? No, not really, because in order to get adaptation, especially like mitochondrial or metabolic adaptation in these like lower threshold motor units, we need to develop a lot of metabolites in them. So hopefully everyone remembers like AMPK, PGC1 alpha, all of that type of stuff. If you're doing a 1 repetition max on, say, I don't know, a deadlift, you're going to send a signal of like the whole AKT-mTOR type of thing to these larger and smaller muscle fibers, but we actually won't break down enough ATP in the smaller muscle fibers in order to like actually get AMPK to be stimulated, and subsequently PGC1 alpha and the electron transport genes, the fatty acid oxidation genes, all of that type of stuff. However, they are being active in some way. So I don't know, like I think that's kind of cool. Next, let's just talk a little bit about the brain. There's different aspects to it. Not a whole lot that I'm really going to want any of you to know or be aware of in here, but we have the cerebrum, which breaks down to the cerebral cortex and the motor cortex. So the cerebral cortex, there's different aspects of this, but this is basically where we organize complex movement, and we have stored like learned experiences within there. Like, I don't know, the fusiform gyrus, for example, is where you store all your faces that you've ever seen. And people who have lesions in the fusiform gyrus can recognize people's voices, but they can't recognize people's faces for whatever reason. Well, not for whatever reason, because that's the part of the brain that works. Sorry, I won't have too many more of those asides because I'm sure everyone's getting bored. Next thing, the motor cortex, and we have a premotor cortex and things, but this is really control and voluntary movement. That's what's going on there. Now, the cerebellum, this has to do much more with fast ballistic movements and also balance in many ways. Now, the vestibular system has to do with balance as well, but the cerebellum has to do a little bit with it too. So it coordinates and monitors complex movement, and it has numerous connections to the motor cortex, brainstem, spinal cord, and a lot to do with fast ballistic, like plyometric, jumping, explosive types of movements. Now here within the brainstem, there's a lot of complex structures that deal with many like metabolic functions and cardiorespiratory control. So this being a little bit deeper, a little bit more protected, we don't want to mess any of this stuff up. There's different parts, the medulla, pons, midbrain, reticular formation. I'm not going to get into that too much. But here, this is functionally where everything is actually stored. Cerebrum, diencephalon, corpus callosum, the part that subdivides the two different hemispheres, the cerebellum, spinal cord. Here's the brainstem, midbrain pons, medulla oblongata, all of that. Now here I'm going to just talk a little bit about concussions. Not much at all. It's really not my area of expertise. But some issues with concussions, there's really just about nothing holding the brain into place. So really the problem isn't so much the impact, but like say if you get hit in the head, the brain bashing against the other end of the skull or being rattled around in there, that causing damage to the central nervous system. And hopefully you remember before, the peripheral nervous system can functionally repair, but the central nervous system, not so much. Now, right now, there's a lot more research coming out about concussions. And actually, in our lab, we have various machines, like this one balance machine, that seems like it might be able to detect if someone is back and ready to play or not. But other than that, I'm not going to talk about sport-related concussions too much. I think your book says some interesting things about it, but I don't know, like here are some associations with it, some different cognitive, emotional, and sleep-related symptoms. So I don't know, you could have a headache, difficulty remembering, you could be sad, emotional, nervous, and have difficulty falling asleep. And, you know, if you control for all sorts of other factors, like if you just got broken up with or whatever, like, yes, you didn't have concussion, but you might experience some of these. Or, I don't know, you might just be feeling under the weather, as I'm sure a lot of people are at the moment. All right, now I'm going to talk about something that is near and dear to my heart, one idea that I've always been fascinated with, and I'm sure most of you are fascinated with it as well. The idea is more or less, why do we get tired? Like, why can't we just sprint marathons, or whenever you're bench pressing something, why do you get tired after 3 times, 4 times, 20 times, whatever you're doing? And there's effectively 2 different competing ideas, and I will say most researchers seem to agree with the peripheral fatigue theory, meaning that there's something outside of your central nervous system that is driving fatigue. But every now and then there is some research that doesn't necessarily indicate, but suggests that central fatigue, or somewhere within the central nervous system, might be related to like fatigue. And I think it kind of makes sense in this more simplistic way that your brain is going to stop your body because, well, your brain more or less knows what you can handle, so it's going to stop you before it's too much. But let's just talk about this really quick. So central fatigue just means higher brain centers and or motor neurons. So there is reduced motor output in your muscles whenever they are fatiguing, so we can measure this through like EMG or different neurotransmitters or things like that. But neurotransmitters or neurotransmitters going on in your central nervous system or peripheral nervous system actually do get somewhat depleted from long amounts of exercise, and this is mostly due to them being made out of various like amino acids and things that we actually like lose them in some way. Now, there's what's called the central governor theory. So central control center regulates exercise performance, and it really just protects against catastrophic problems. So making it to where you don't exercise yourself into oblivion or into death. Now, a lot more research needs to be done on this mechanism. Now, something here, this is probably mostly related to endurance exercise, but a lot of time, what is this called? Transcranial stimulation, like TMS, transcranial magnetic stimulation. I believe that's what it is. like if an individual is exercising on a bike, this is typically how it's done in a laboratory setting. Once they get to the point where they're really fatigued, you'll put a particular, like, I don't know, electromagnetic coil on, like, right over their motor cortex, and then that's going to send a signal, like an electrical signal, that will frequently increase how long an individual can persist on some type of endurance task. So I don't know. I think that that is abundantly interesting. Here is another thing. So here are just a couple of things. TRP, that's tryptophan, so the amino acid associated with having to go to sleep, which turkey actually isn't very high in tryptophan, so don't be ignorant, don't say that. There's a lot of other foods much higher in tryptophan. The reason that we get sleepy after Thanksgiving is because you eat so much, and then all of the blood pools in your gut, it goes away from everywhere else, and then you just have to go to sleep to digest and deal with it. But here what's going on. TRP, that's tryptophan. 5-HT, that is serotonin. So 5-HTP is actually tryptophan. But within the brain, whenever we get a little too much tryptophan or overall like serotonin in the brain, then that typically sends a signal for fatigue or to stop persisting. Now, an interesting thing about the branched chain amino acids, leucine, isoleucine, and valine, if an individual supplements with these, there is a little bit of research that there is less 5-HT or serotonin formation. So if an individual is going on super long endurance, like exercise types of things, like ultramarathons and things like that, you might actually delay fatigue by ingesting branched chain amino acids via this mechanism, because branched chain amino acids and tryptophan similar enough in structure to where they can bind to these different receptors and either promote serotonin production or inhibit serotonin production. So branched chain amino acids would seem to inhibit serotonin production. So yeah, I don't know, like I think that that's kind of cool. All right, let's get into just a couple of reflexes, and I promise we're almost done. Well, I'm sure you can look on the video and see how accurate that statement is. So motor functions of the spinal cord. So we have withdrawal reflexes, other flavors of reflexes, the cross-extensor reflex, and we have something called spinal tuning. Spinal tuning is something that, like, I do want you to know about. So effectively, we get a rough draft of a motor function, like say whatever type of contraction, or numerous muscle contractions together to, like, say, throw a curveball or whatever. We get that information from higher level brain centers, and I'm going to show you kind of where this is coming from. And then it's going to go on down through your body, and your spine is effectively going to tune it to get it exactly right. And there are various studies on, like, say, dart throwers, that within the signal of how they throw a dart, expert dart throwers actually have more variance in their upstream motor patterns, meaning in, say, I don't know, like their shoulder or whatever, like their elbow, but less variance in what they actually do with, like, say, their wrist. Like they make things a little bit more exact. They pay attention to the parts that are a little bit more important for whatever motor task that they're doing. So here, let's just look at a couple of these. So withdrawal reflex. So reflex contraction of skeletal muscle. This occurs in response to some type of sensory input, and it's not dependent on higher brain centers, meaning it just stays in the spinal column. So here you could read about pathways of neural reflex. I'm not really going to talk about it. Now there is a reciprocal inhibition, meaning EPSPs two muscles to withdraw from the stimulus, and IPSPs to antagonistic muscles. So I'm going to show you this cross-extensor reflex, and that's hopefully going to make sense here in a little bit. So how this works, say we hit a pin with our finger. So over here, these flexors are stimulated, so we get a lot of EPSPs here to contract, and then these extensors are inhibited, meaning we get a lot of IPSP signals here. Now this cross-extensor reflex means that just the opposite is going to happen on the other limb, meaning that these flexors are going to be inhibited and these extensors are going to be stimulated. So kind of the idea behind this is if this wasn't a pin, but it was something else, like, I don't know, a person hitting you or something, that there's effectively going to be, like, you're going to retract one aspect of your body and you're going to send out the other aspect of your body to push whatever that, I don't know, negative stimulus is away. So, like, say, I don't know, like if someone's trying to tickle you and they're tickling one of your arms, you're going to retract that arm and then you're going to hit them with your other arm. At least that's what I would do. But that's neither here nor there. Now let's just look at a little bit of control of motor functions. So we have subcortical and cortical areas. So subcortical just means deeper brain centers. Cortical means like more on the surface of your brain. Now there's a rough draft of an idea there, and really I kind of want us to think about the like motor cortex. Now we then send a signal to the cerebellum or basal ganglia, and really the main thing here is that we have fast movements traveling through the cerebellum and more slow deliberate movements going through the basal ganglia. And then motor cortex through the thalamus. So this forwards more information, then we have spinal tuning happening, and then we have some feedback from muscle receptors, proprioceptors, chemoreceptors, all sorts of things like that. So here is more or less like a step-by-step way of looking at it. So it effectively is just going through this region. I'm not really super married to having y'all know too much about this. It's I do find it fairly interesting, but you know, just like look through it a little bit. Now, almost done. Whenever we think about the autonomic nervous system, we think about the sympathetic fight or flight and the parasympathetic division. And the main things that I want you to know is that the sympathetic fight or flight is sending out primarily the norepinephrine neurotransmitter, whereas the parasympathetic division of the autonomic nervous system is sending out acetylcholine. So that's really the main things that I want you to know. So like norepinephrine excites whatever organ, and the acetylcholine inhibits it. So yeah, like we could get into acetylcholine esterase and how there's different inhibitors of that, but I'm not going to talk about it too much. Here, just generally, like what's going on, there are a little bit of differences with, like here, cranial parasympathetic, right there, ACH, sacral parasympathetic, ACH, which just means acetylcholine. Here's the sympathetic ones, which are, you know, like more in this thoracic lumbar region. Just another term I want you to be aware of, acetylcholine, choline, so cholinergic, and then norepinephrine, adrenergic. So remember that norepinephrine and epinephrine bind to adrenergic receptors. So adrenergic, that's really how that works. Not a whole bunch that I want you to know here. I don't want you to like recall the preganglionic neurotransmitter and the postganglionic one. That's a little bit beyond what I care for any of us to know. But that's really about it. And I will see you in the next lecture.